Our next series of podcasts are going to deal with some basic concepts in genetics. This first one will deal with genetic variation. If you consider genetic diseases in relation to single gene mutations, from the online Mendelian inheritance in man, we know that there are something like 19,000 single gene traits that are defined in humans. There are more than 18,000 of these single gene traits on autosomes, perhaps 1,000 or more on the X chromosome, with maybe only 60 or so on the Y chromosome. If we consider a single gene trait, we're thinking of a monogenic trait or a Mendelian trait, and the behavior of these are exactly what you may have learned about in high school or college biology classes. So in this matrix or Punnett square, we can say that big A is dominant to little a, which is recessive. If you work this Punnett square, you can see that there are 3 genotypes and 2 phenotypes. So the 3 genotypes would be big A, big A, big A, little a, and little a, little a. The 2 phenotypes would be those phenotypes that are expressed by having the dominant genes or the recessive genes. This is the easy part of genetics because many, if not most, human diseases have genetic components that are not related just to single gene traits, and even those that are related to single gene traits may have many different mutations associated with them. So for example, and I'll talk about this a little bit later, but cystic fibrosis is an autosomal recessive condition, but there are more than 2,000 known mutations associated with the gene that causes cystic fibrosis. If you look at 2 genes, the picture is much more complicated. You can see the table on the left showing the genotype ratio with 2 genes. There would be 9 genotypes, and you can see the phenotype ratio of 9 to 3 to 3 to 1. Now in this course I'm not going to ask you to calculate things like this, but just it gives you an idea of how complex things get when you talk about more than one gene. For learning issues for this next series of podcasts, you should be able to recall some basics from biochemistry that relate to genetic variation and mutations. You should be able to distinguish between gene frequencies and genotype frequencies. You should be able to use the Hardy-Weinberg relationship and or Punnett square analysis to solve basic genetic problems. You should be able to determine recurrence risk from simple scenarios that you might be presented with. You should be able to work simple pedigrees to make genetic diagnoses. You should be able to distinguish between autosomal and sex-linked inheritance. And you should be able to understand implications of germline mosaicism, penetrance, variable expression, locus heterogeneity, and imprinting. 2 principles from basic genetics that I hope you remember. Segregation: genes occur in pairs and only one member of a gene pair is transmitted to a gamete. Why do we say that genes occur in pairs? Well, we understand that we have homologous chromosomes, one from mother, one from father, and likewise there are homologous genes. The gene that occurs for a given trait would come from each parent. We also talk about independent assortment. Genes at different loci are transmitted independently of each other, and if the genes reside on different chromosomes, we know the chromosomes assort independently during meiosis 1, and you remember the concept of 2 to the 23rd for the number of possible gametes that can occur as a result of meiosis. Now, just a thought question. Can we say that independent assortment does not occur? Well, if you think about genes on different chromosomes, then you know that the chromosomes assort independently of each other, but you also know that if genes are linked very close together, they can't cross over, and Therefore, those genes don't independently assort from each other if they're linked together on a chromosome. These principles are key not only to understanding heritability of single gene traits, but they're basic to understanding why meiosis works as it does and how one can use linkage analysis to trace disease patterns within families or within larger groups of people. Genotype is an individual's genetic constitution at a locus. The phenotype is what is physically observed or what is clinically observed. Genotypes don't necessarily correspond to unique phenotypes, and individuals with different genotypes may have the same phenotype. So, for example, cystic fibrosis is a single gene autosomal recessive condition. Only individuals who are homozygous recessive, a would be affected by cystic fibrosis. Individuals who have the homozygous dominant genotype, for example, big A, and those with the heterozygous genotype, big A, would show the same phenotype, and they would be normal, although individuals that are heterozygous, big A, would be considered carriers for a disease like cystic fibrosis. Because there are almost 2,000 known mutations in the gene that causes cystic fibrosis, the disease itself can manifest with very mild to very severe symptoms, and we'll have more to say about that later. The same genotype might also produce different phenotypes within different environments. PKU, for example, is another single gene autosomal recessive condition. It can lead to severe mental retardation, but you can avoid all the adverse phenotypic effects of PKU by placing babies on low phenylalanine diets within a month of birth. We know that humans display fairly substantial genetic variation, and this is reflected in traits like height, blood pressure, skin color, or different disease states. Genetic variation arises as a result of mutation. If the variation is in the germline, that variation can be transmitted from one generation to the next. But if that variation occurs in somatic cells, that might lead to cancer. We also know that variation arises due to multifactorial inheritance, and we'll have a lot to say about multifactorial inheritance throughout our course, but that would relate to genes interacting with the environment, and especially when you can't attribute traits just to one gene. And this is just a diagrammatic example just to give you a sense of what a single gene trait might look like and how you can easily see how it would be expressed, how if you had a digenic trait, how it would be expressed, and then if you had multifactorial inheritance. So for example, stature in humans is determined by a number of different genes and environmental factors, and so if you look at stature from short to tall, you can see how the population frequency arrays itself almost on a normal distribution, and this would reflect the interactions of genes and environmental factors on a complex trait like stature. These figures give you more of a pictorial view, looking at genetic variation, and it's looking at genotype numbers versus the number of genes. For simple Mendelian crosses, these grids represent genotypes, and they would assume no variation within the genes. I don't expect you to make a big deal of this, but it just gives you an idea of how complex things get. If you talk about a single gene, we've already mentioned 3 genotypes, 2 phenotypes. If you talk about 2 genes determining a trait, then you know there are 9 genotypes with 4 phenotypes, but look at how complicated it gets if you have something like 5 genes determining a trait. Look at the number of genotypes, look at the number of potential phenotypes, and this is not even considering how the genes interact with each other or with the environment. This is just simple Mendelian traits. things to recall from your studies of biochemistry: there are molecular consequences to mutation. You've talked about gain or loss of function mutations. You've talked about haploinsufficiency. You usually see this in autosomal dominance, like in familial hypercholesterolemia, where maybe 50% of the gene product is not enough for normal function. You've also talked about clinical consequences of mutations, for example, with respect to hemoglobin disorders. You've talked about causes of mutations and mechanisms for repair, and you've talked about various molecular biology methods. So these are all things you should keep in mind as you approach your study of genetics. Now, types of mutations with respect to translation or protein synthesis that might lead to genetic variation. We can talk about base pair substitution or point mutations. These can be missense mutations where there's a change in amino acid sequence. There can be nonsense mutations where there's premature chain termination, or there may be silent mutations where you see no effect because of the mutation, perhaps because the genetic code is redundant. We can talk about deletions and insertions that can lead to frame shift, where there may be a change of amino acid sequence, or you may add or lose an amino acid. You talk about duplications of entire genes, and that might lead to dosage effects. You've of course talked about insertion of mobile genetic elements, and you've also talked about mutations that might occur in non-translated sequences. So perhaps there's mutations in promoter or enhancer sequences that relate to how genes are expressed. You've talked about splice site mutations, especially in donor or acceptor junctions. And you may talk about mutations in ribosomal DNA sequences. These are usually non-consequential because the ribosomal genes are middle repetitive genes. In other words, they're redundant. Now, a gene is the fundamental unit of heredity. You already know this. DNA sequence, which is associated with regulatory regions, transcribed regions, or other functional sequence regions. All of those DNA sequences would be part of a gene. Now, as a result of mutations, a gene can differ among different individuals with respect to DNA sequence. The differences in DNA sequence represent the alleles, and so the alleles are alternative forms of genes, and you'll find alleles on a maternal chromosome and a paternal chromosome. The locus is actually the gene's location on a chromosome. This is just an example. I think you may have talked about this in your studies of biochemistry, but you know that patients have different alleles at the beta globin gene locus on chromosome 11. So a homozygous individual, the patient would have the same allele at the beta globin locus on both members of chromosome pair 11, whereas if the person was heterozygous, the patient would have different alleles at the beta globin locus on the maternal versus the paternal chromosome. The alleles that are present at a given locus would be the patient's genotype. Now, we talked about cystic fibrosis. That's an autosomal recessive condition. So here I'll just assume that big A is the wild type version of the allele, little a is the mutant version of the allele. The gene responsible for cystic fibrosis, it codes for the cystic fibrosis transmembrane conductance regulator. This is a transmembrane protein that regulates movement of chloride and hence water across cell membranes. If you look at the relationship between genotype and phenotype with respect to disease outcome, three factors might contribute to severity and variability of a disease like cystic fibrosis. First would be the patient's genotype. In other words, the patient's specific pair of cystic fibrosis mutations. There may be other genetic factors, like whether there are cis or trans variants that might affect intron splicing, and of course there may be environmental factors that might include diet and or therapy. And as we said, there are almost 2,000 known mutations for cystic fibrosis. An individual that's wild type or homozygous would not be affected, obviously. here's an individual that's heterozygous, but this person is a carrier because one of the alleles is the wild type, the other allele is this mutation, so the so-called delta F508. This happens to be a phenylalanine deletion at position 508. You don't have to know that, but this person is a carrier. They're not affected. Here's another carrier. Again, not affected because one of the alleles is a mutated allele, but the other allele is the wild type allele. On the other hand, here is a patient that has two mutant alleles. This would be a homozygous patient. This patient would happen to have severe lung disease and pancreatic insufficiency. Here's another homozygous individual. This individual has a bilateral absence of the vas deferens, but otherwise might not have as many severe conditions. These are homozygous recessive mutations, but we can also talk about individuals that are compound heterozygotes. That, in other words, they have two different mutations in the CF gene. And compound heterozygotes reflect diversity of mutations in an autosomal recessive disorder. And knowing the different recessive mutations might be useful in relating to clinical management of people with cystic fibrosis. I'm not going to ask you at this point in the course to know what these different mutations stand for or even know what the symptoms are. Now we can talk about polymorphisms and mutations, and in human genetics, mutations are usually defined as changes in DNA sequence that result in genetic diseases. These generally occur at relatively infrequent rates in a population, so mutations are considered to be rare. In human genetics, polymorphisms are recognized when two or more clearly different phenotypes would exist in the same population. Polymorphisms are common in nature, and these would relate to biodiversity, to genetic variation, and to things like adaptation. And polymorphisms would be driven by evolution to retain variability in populations that live in varied environments. So, for example, sexual dimorphisms, mimicry in insects, we talk about the various human blood types. These are all polymorphisms. You can see polymorphisms in, for example, in male versus female birds, but we know about sexual dimorphisms even in humans. I don't need to go over these for you, but you can also see polymorphisms in cardiac enzymes. And I'm not making a big deal here. I just want you to be aware that we know that polymorphisms exist in all kinds of different human traits. Now, when two or more alleles at a given locus have frequencies that exceed 1% in a population, they're said to be polymorphic. Polymorphisms can be recognized at frequencies of less than 1%, so polymorphisms are known to influence the risks for contracting complex diseases like diabetes or heart disease, and the distinction between mutations and polymorphisms in human genetics is becoming more and more blurred.